My name is Navaithani Mahendran, and I'm a third-year master's student with the School of Interdisciplinary Health Sciences at the University of Ottawa. I'm completing my master's thesis project under the supervision of Dr. Anne Conkle, and our project is investigating the interaction between gestational immune activation and environmental enrichment during pregnancy on ER alpha and IBA1 counts in the postpartum maternal rat hippocampus. Pregnancy is understood to be a period of plasticity to prepare for offspring care that is largely mediated by enrichment from exposure to new stimuli. Changes occur with the maternal immune system, which includes a shift from pro-inflammatory to anti-inflammatory responses. This change is also observed at the neuroimmune level, as studies have observed a significant reduction in microglial density during late pregnancy and early to mid-postpartum period. Neuroendocrine changes are also observed through fluctuations in the level of gonadal steroid and pituitary hormones, which help with the progression of pregnancy and post-pregnant state. The estrogen signaling pathway is of interest, as there is some support for elevated estrogen receptor alpha expression in lactating females, which is also associated with higher maternal care behaviors. Moreover, a complete knockout of alpha in rodent studies have resulted in deficits to maternal care. These changes are not observed with beta. Some studies have found a reduction in alpha expression in brain regions that are directly involved in maternal care. Lastly, neuroanatomical changes are also observed through a reduction in cortical thickness and cortical volume, a marked reduction in gray matter volume, and increase in ventricular size. Reductions in gray matter volume in regions such as the hippocampus overlap with structures that are necessary for offspring interactions. The maternal hippocampus is of particular note because it is implicated in indirect maternal care behaviors. Essentially, because the hippocampus is involved in important cognitive functions such as learning and memory, it plays an important role in mediating behaviors that support offspring care. Infections are a prevalent risk factor during pregnancy. An acute immune challenge during this sensitive period has the potential to compromise key neuroimmune, neuroendocrine, and neuroanatomical processes that are implicated in direct and indirect maternal care behaviors. An immune challenge in the mother during pregnancy is termed gestational immune activation, and its effects on the maternal brain are not well understood. There are potential protective factors such as environmental enrichment, which has been found to act as a buffer for inflammatory responses through providing resistance to inflammation and increased expression of neurotrophic factors. With this, our research questions are: 1. Does an immune challenge interact with the housing environment to influence the number of alpha positive cells and/or IBA1 positive cells in the CA3 region of the postpartum rat hippocampus? And 2, do any of these treatments individually have an impact on the number of alpha positive cells and/or IBA1 positive cells in the CA3 region of the rat hippocampus? To investigate this, 42 Sprague-Dawley rat dams were randomly housed in one of three housing conditions following breeding: standard housing, social enrichment, or environmental enrichment. Then, on gestational day 11, the mothers were treated with either an intraperitoneal lipopolysaccharide or saline injection. Lipopolysaccharide is a component of bacterial cell wall, and it is a mimetic for bacterial infection. The day the pups are delivered is noted as parturition day P1. On parturition day 22, the mothers are sacrificed and brains are collected. The collected brains are fixed and later sectioned for immunohistochemistry procedures. Immunohistochemistry is used to target estrogen receptor alpha and IBA1 positive cells. Estrogen receptor alpha is present in neurons involved in estrogen signaling. The number of estrogen receptor positive cells will act as a proxy for estrogen signaling levels. IBA1, or ionized calcium binding adapter molecule, is a microglia-specific calcium binding protein. IBA1 is present in activated microglia. The number of IBA1 positive cells will act as a proxy for neuroimmune activity. Following immunohistochemistry, sections are mounted on slides, and microscopy and cell counts are completed based on Bregma assignments for each section. Results from completed cell counts demonstrate that dams in different housing conditions did not significantly differ on number of ER-alpha positive cells. This can be seen in Figure A. Moreover, housing and treatment did not interact to affect the number of ER-alpha positive cells. However, as you can see in Figure C, dams treated with LPS had significantly reduced ER-alpha positive cells compared to dams treated with saline. In Figure B, we can see that dams in different housing conditions did not significantly differ on the number of IBA1 positive cells. Housing and LPS did not interact to affect the number of IBA1 positive cells as well. And in Figure D, we can see that dams did not significantly differ on the number of IBA1 positive cells based on treatment. The current data is based on 32 and 20 dams of a total of 42 dams for the ER-alpha and IBA1 analyses respectively. Thus, definitive conclusions about trends and significance cannot be made at this time. Overall, the current data suggests a potential trend of reduced ER-alpha positive cells in animals treated with LPS, as seen in Figure C. These findings are in line with other studies in our lab which have found a reduction in ER-alpha positive cells in non-hippocampal regions such as the BNST. These preliminary data also appear to show that environmentally enriched animals may not experience additional protective effects from the LPS treatment condition as expected. The next steps for this project include investigating whether an immune challenge in housing conditions individually influence and/or interact to change a number of ER-alpha positive cells and/or IBA1 positive cells in the other layers of the postpartum maternal hippocampus. This includes the CA1 and dentate gyrus of the hippocampus. This project will allow future research to examine the effects of infections in the context of postpartum maternal mental health. This will require assistance from epidemiology to collect information on the incidence of infections in pregnant women, investigate features of infections that are most detrimental, and this includes the type of pathogen, the trimester of infection, the length of infection, and the need for hospitalization. Future research will also inform health policies to provide more robust mental health care.